Yeah, anything I mentioned could potentially be on there. So yeah, I would definitely take notes on it. All right. Well, getting back to this diagram here. I guess the point I was making there is that through the interfering RNA interference, the mRNAs can be selectively degraded, or at least the ribosome selectively blocked from any particular mRNA. Let's say that the ribosome does manage to translate the mRNA, and so you get the actual protein being made. Even post-translationally, even after the protein has been made, there are opportunities for the cell to regulate the activity of that protein, and so that also counts as differential gene expression. Not all proteins, but many proteins are initially made with what you can call an inhibiting domain. In this diagram, the blue-colored part of the protein you can think of as the functioning protein. This is actually insulin hormone, which is a protein, but think of that as the functioning part of the protein. Well, many proteins are first made with an inhibiting domain that essentially covers up the active site of the protein. It just blocks off the active site, like like on a safety razor, they have a plastic thing that fits over the top of the razor to block the actual blade part of it. And so the cell has enzymes that find these inhibiting domains and chop them off so that the protein becomes functional. And that's, for instance, what happens to the insulin that your body makes. Before it becomes functional, that domain has to be removed. Well, by regulating the activity of these enzymes that cleave off the inhibiting domain, the cell can therefore regulate the activity of the protein itself. So yet more opportunities for differential gene expression. Another type of differential gene expression that can happen at the protein level is this. We've talked about how certain proteins only become activated when they are phosphorylated, meaning that phosphate groups get attached to some of their amino acids. So like this protein is normally in an inactive state, but if a phosphate group is attached to maybe one of its tyrosine amino acids, it folds itself into a slightly different conformation, slightly different structure, and then becomes active. Who remembers the name of the enzymes that add phosphates to amino acids? Begins with a K. Tyrosine kinases. And so it's the activity of the kinase enzymes inside the cell that can serve to regulate the activity of other proteins inside the cell. Like that. All right, and lastly, imagine that the protein is made and it has become active, so the protein is actually carrying out its function inside the cell. Another way to regulate that protein is to limit the lifespan of that protein, because just like I mentioned with messenger RNAs, cells don't want proteins hanging around forever, because just because a cell might need an enzyme at this moment, it may not want that enzyme to be functioning at the next moment. And so the cells have ways of getting rid of proteins. Proteins have finite lifespans in the cell. And here's how that works. There's a small protein called ubiquitin. I don't know exactly how many amino acids it is, but it's a pretty small protein. And the cell attaches the ubiquitin protein to the proteins that it wants to get rid of. The ubiquitin is essentially a tag or a flag that says, let's get rid of this protein. It's sort of a destruction tag for protein. So the protein that's to be degraded gets ubiquinated. And then there's an organelle inside the cytoplasm called the proteasome that's like a shredder for ubiquinated proteins. The proteins that have been tagged with ubiquitin go inside the proteasome and it tears them apart, eventually down to the amino acid level. So the amino acids can be reused to make new proteins. Well, the cells are able to selectively decide which proteins they want to add ubiquitin to. So if the cell wants to downregulate expression of a fully functioning protein, what can it do? The cell can do is selectively ubiquinate that protein, and so that slates that protein for destruction by the proteasome more than other proteins, and that's differential gene expression. And it's so in some cases you find the ubiquitins are added to proteins that the cell just wants to downregulate the expression of. What they also find is this, that the cell will also add ubiquitins to denatured proteins. Remember, denatured ones are where the protein has folded in the wrong shape. Maybe the cell got too hot and the heat caused it to denature, or maybe there was a mutation in that gene, so the protein is now folded into the wrong shape. The cell is somehow able to identify denatured proteins floating around in the cytoplasm, and those proteins tend to be selectively ubiquinated, so they'll be destroyed. And viral proteins also, the cells seem to be able to figure out capsid proteins and reverse transcriptase proteins and other proteins that come from viruses, and those ones are also selectively ubiquitinated, so the cell can get rid of them. Okay, so I guess the big picture of today's lecture so far is that, yeah, most of the differential gene expression does take place at the transcriptional level, but there's lots of other areas where it can take place also. Processing the pre-mRNA, having the ribosomes bind to the messenger RNA, or the lifetime of the messenger RNA, or even after the protein's made, there are ways of controlling its activity or tagging it for destruction. Good stuff? All right, well, let's see. So we're still in this genomes chapter, but I want to start a different area within the genomes chapter, and we're essentially done with differential gene expression. Before the 1990s, they didn't really have a clear idea of what the human genome was all about. Remember, the genome means all the DNA in an organism, so it includes all the genes and DNA that's also not genes, DNA that doesn't encode for proteins. And certainly human genes had been sequenced before the 1990s, but it was essentially just individual genes. Nobody had sort of a global picture of what all the genes we had were and all the non-protein encoding DNA was all about. And then in the early 1990s, they decided this would be a very useful thing to know, the complete human genome. And so an international scientific project called the Human Genome Project was started, where they essentially decided we're going to sequence all the human genome, all of 23 types of chromosomes that humans have, chromosomes 1 through 23. That was a tremendous amount of sequencing to be done. If you add up all the chromosomes, it's around 3 billion base pairs all told. And the sequencing methods at that time were pretty slow. And so what they had to do is divide it among many institutions, many research institutions throughout the world. Here's a graph that shows how they allotted different sequencing to different institutions. And I don't know exactly how they picked who would do what, but maybe they said, hey, Sanger Center, you guys do chromosomes 1 through 7, and Whitehead Institute, wherever that is, you guys do chromosomes 8 through 10, and you guys do these chromosomes, and you guys do these chromosomes. But somehow they decided who would be sequencing what chromosomes, so that the entire genome will be covered. And they started the actual sequencing in 1999, and finished it just about 11 years ago. In 2001. So it took about 2 years of solid sequencing. These are some of the sequencing machines that they just had running day and night to get it all sequenced. And as they were doing the sequencing for the Human Genome Project, the technology of how you could sequence DNA got better and better. In other words, the methods that they started with were not the methods that they ended with. And kind of interestingly, a private company invented a super fast sequencing method. And so even though this private company started later, they almost finished in a dead heat with the Human Genome Project as far as sequencing all the DNA. But anyway, in 2001, they had said, okay, we've completed it now. They've sequenced the entire human genome. And there were a lot of surprises, unexpected things that they found in the results. One of them was how few genes human beings had. They found out it was around 25,000 protein encoding genes in human beings. And the reason why that was surprising is they thought it was closer to 100,000. They thought it would be closer to 100,000. One of the reasons why they thought it would be closer to 100,000 is because they find a lot more proteins in human beings than just 25,000. So who can tell me how can we get more proteins than we have genes? For what? Or genes can make more than one protein by what method? Alternative splicing. Yeah. So they think that the vast majority of our 25,000 genes can do alternative splicing. So we probably have at least 50,000 different proteins in us, and maybe close to 100,000. But 25,000 protein encoding genes is what they found. And since they've done the human genome, they've sequenced the genomes of other species also. And you don't have to memorize any of these numbers, but just in case you're curious, a typical bacteria might have around 3,000 or 4,000 genes. Yeast, which are eukaryotes, but very simple single-celled eukaryotes in the fungi kingdom, around 6,000 genes. Insects like fruit flies, about 13,000, 18,000 for a nematode worm, which is a very small worm. Plants, in many cases, have more genes than we do, which I found a little bit insulting at first, but then I remembered that, well, plants have to do photosynthesis and we don't, so they need a whole bunch of extra genes that we don't have. But the biggest surprise was not that we had only 25,000 genes. The biggest surprise was this: how little of our genome, how little of our DNA was actually genes. The encoding the proteins that we need for survival. It turns out that only around 25% of the genome is actually protein encoding genes. And it gets even more mind-boggling than that, because when we say the word gene, people include the introns in the gene, which don't really encode any proteins, the regulatory sequences like the promoters and the enhancers, which don't really encode any proteins. They just regulate the gene. And other non-coding parts like the 5' untranslated region and the 3' untranslated region. So a lot of these genes aren't even really protein encoding codons. If you look at, say, well, how much of it is actually protein encoding codons, like the exons themselves, it's really small, like only about 1 or 2% of the human genome actually encodes for amino acids. And throughout this course, I've been saying, the purpose of the DNA is to encode proteins, but actually only about 1/100th of it is actually in the business of having codons for encoding the amino acids. So it sort of begs the question, what is the rest of all this stuff? Well, the other parts of your genome, some of them are non-functional genes. There's some parts of it called large duplications, other parts called tandem repeats, and other parts called transposable elements. And notice the transposable elements actually makes up the largest portion of it. Well, I'll tell you what, let's stop here. When we come back on Friday, I'll finish off what the other parts of the genome are.